Good evening, everybody. This is Benjamin. Tonight, I want to discuss this picture where this picture speaks 1,000 words. This is a description of a synapse, that is the communication between two neurons. Now, the space here is called the synapse, and a lot of things happens between a part of the neuron and another part of the neuron communicating. This part could be different. This could be a dendrite of a neuron, where this could be a terminal board, or vice versa, or a cell body of a neuron and a terminal board. But we're not going to talk about all that. I want to basically discuss the synapse, the point of communication between two neurons. I gave the term neurocommunication. What happens? How do neurons communicate? I'm going to be describing all that with this picture, because this picture is going to speak 1,000 words. When there is a change in the membrane of a neuron, it elicits an action potential. Now, the action potential, when it happens, that is, when you have change in polarity of neurons. Normally we could have all positive in the neuron in the outside membrane, while the inside means could translate to all negative. When there is a stimulus, there's a kind of change in the membrane in which some positive from outside goes inside, and that elicits an action potential. Now, there's an influx of calcium ion, that's Ca2 plus here. When there's an influx of calcium ion, this is what happens. You see these vehicles, they are packed with neurotransmitters. Neurotransmitters are communicating chemicals between neurons. So we could have acetylcholine, norepinephrine, serotonin, dopamine. Those are chemicals in which neurons use in communicating. So when there is a stimulus and there's an action potential, the influx of calcium ion is going to move this vesicle packed with neurotransmitter close to the membrane of the terminal board of the presynaptic neuron. The neuron before the synapse is the presynaptic neuron, while apparently the neurons after the synapse is the postsynaptic neuron. When this vesicle moves down, there's a kind of a degrading of this vesicle with the content in there through a process called exocytosis. And what we have is the release of neurochemicals or neurotransmitters, which I said earlier could be dopamine, acetylcholine, epinephrine. And when these vesicles, when these substances are released, the chemicals are released, they bind to a receptor at the membrane of the postsynaptic neuron. This is the postsynaptic neuron, and there's the membrane there. So you have the receptor binding to the neurochemicals or neurotransmitters. This will not elicit the desired action potential in this place and this postsynaptic neuron, and it continues like that. Now, there are some interesting factors that we need to consider here. It's an interesting phenomenon, and that's these neurotransmitters, they don't stay all throughout in the synaptic space. Sometimes they are broken down by the lysa. You have the likes of monoamine oxidase, which breaks down dopamine into some biodegradables or degradables that are taken back into the presynaptic neuron for repackaging, so that there could be availability of neurotransmitter in vesicles for the next action. Now, if you see this place, you can see, like we are taught for years, that neurons actually have a space between them called the synapse. Apparently that is true, but actually there is a connection between the neurexin and the neurolignin. Now, these are kind of molecules, some molecules that are bounded to the membrane of both the presynaptic membrane and the postsynaptic membrane of the neurons. They communicate. Now, we have receptors coding for all this, the genes coding for all these receptors or molecules or membrane substances. And now, defect in the receptor or in the genes coding for neurolignin or neurexin has been implicated in some form of developmental disorders. You could do so much on, or you could do some researches on that too. So I think I have been able to explain this picture relating to speaking 1,000 words, where there's action potential, influx of calcium, ion, the movement of the vesicles down to the membrane of the inner membrane of the presynaptic neurons, and there's degradation of the membrane, and there's release through the process of exocytosis of the neurotransmitters. The neurotransmitters bind to the receptor on the postsynaptic membrane, and action potential is elicited, and there is a kind of communication between some molecules or receptors between the presynaptic membrane and the postsynaptic membrane of the neurons, and these molecules are called neurexin and neuroligin, which are implicated in which, if we have default in the genes coding for these proteins or these receptors, are implicated in developmental disorders in children. And when I said the other time that when neurotransmitters are released in the synapse, they are broken down by lysa of the neurotransmitter. Example is monoamine oxidase that breaks down dopamine, and we also have acetylcholine esterase which break down acetylcholine. And we should also know that there's lysa, there's molecules that break down, or these chemicals that break down neurotransmitters across the synapse, could have some clinical factors in which they are hyperactive or they're less functional. You could have some clinical features or clinical presentation in neurons, or between neurons and muscles. And we also talked about when these molecules, the neurotransmitters, have elicited their action on the receptors, they're broken down and they are reuptaken back into the neurons for further repackaging. And there could also be clinical manifestation, especially in neurodegenerative disorders like Parkinson's disease, of repackaging of dopamine. And issues relating to addiction and drug addiction, has to do with the actions of the drugs taken on the neurotransmitter at the synapse. So I hope I've been able to explain one or two things for your understanding. Thank you. See you some other time.